Hi, and welcome to today's Plant Cell webinar. My name is Jason Padilla, and I am your technical host for today's webinar. Today's webinar features Sachiko Toma-Fukai, Yingshan Li, Yongfei Fang, and it's facilitated by our today's hosts, Pablo Manavella and Sara Gomollon. Today's webinar celebrates the recent focus issue of the Plant Cell Focus issue on RNA biology, edited by Mike Axtell, Andrea Barta, Xuemei Chen, Nan Eckardt, Gregory, Hongwei Guo, Pablo Manavella, Blake Meyers, and Becky Mosher. This webinar is recorded and will be posted on Plante YouTube channel, so be sure to follow our YouTube channel to receive notification when new content is posted, and check out our other talks. I would like to give a special thank you to all our ASPB members who are attending today. Your ASPB membership dues help support and making this webinar possible. If you're not a member yet, you can join today and use the discount code %10 and to receive a 10% discount on registration. You can learn more about ASPB and the opportunities we provide at aspb.org. Before we get started, I'd like to go over a few housekeeping items to make sure that you get the most out of your experience by attending today's webinar. If you're experiencing technical issue, please let us know about those using the chat box or by emailing me at jpadilla@aspb.org. If you have questions for our guest speakers, please add them to the Zoom Q&A box. Our moderators will be sharing them with our speakers. If you have trouble connecting or need to leave the webinar early, again, this webinar is recorded and will be posted on our YouTube channel. All right, now I want to briefly introduce our host for today's webinar, Pablo Manavella. Pablo is an associate professor at the School of Biochemistry and Biotechnology at Universidad Nacional del Litoral in Santa Fe, Argentina. And he's also an independent researcher at the National Research Council, or CONICET. Take it away, Pablo.
Thank you for the introduction, Jason. I'm going to share my screen now. Okay, hold on. You all guys see it? Okay, first of all, I want to thank all the authors and the editor that make this special issue, focus issue possible. It was a great experience. We have been working with it for almost a year now. And we all know that if DNA is the playbook of life, then RNA will be the company of actors that bring life into being. And for the last years, we already learned it and know that RNA is likely the original molecule that gave rise to DNA. Actually, it's also probably the first enzymes and created the first protein that allowed the flow of genetic information that we know as a central dogma. Although the central dogma is still holds true, we now know that RNA play like a critical step that just go a little bit beyond that central dogma that we learned it in school. So we now know that RNA has like different regulatory function, epigenetic activity, and so on and so forth. Especially during the last year and the pandemic, we learned that RNA, we learned a lot about RNA, and actually probably my grandmother now knows a lot of RNA just by watching TV shows and news, reading newspaper. So RNA became a central molecule in our world, and we know that it's not just a side product of DNA, like an intermediate molecule going from DNA to protein. So in this focus issue, we celebrate that, and we highlight many of the forms and function of RNA in plants. This special issue, this focus issue include 10 reviews and 7 research articles. And something that I really like to highlight is the cover art that was designed by Nicolas Inquirani, It's based on Da Vinci's Atlanticus Codex Atlanticus. What makes this cover special for me, at least, is that Nicolas is my nephew. So it is kind of special for me to see it. If you have time and willing to do it, you can even find some Easter eggs on Da Vinci's handwriting in the cover. If you find them, let me know. Not now. Now you have to pay attention to the webinar. Okay, so this webinar, as I told you, featured 10 review articles. The first one is a vignette style review that was written by myself and another 11 groups, expert in different fields of the RNA biology. And we discussed the present and discussed compelling questions that are still open in the plant RNA biology field and the future perspective. You can read them. You can read the vignette that you prefer. You don't need to read the whole article, but I really recommend you go through it. Then Asman and colleague review how RNA structure inform function, drawing an analogy on RNA that can be relatively static, like a rock, having catalytic functions like cutting bonds, like scissors, and can adopt a lot of functional shape like papers. Sebastian Marquardt, Ezequiel Patricio, and myself also write a review article exploring the crosstalk between transcription and RNA maturation, just kind of blurring the bounds between the step-by-step process that we know, or we will teach about. Then Julieta Mateos and Dorothy Steiger discuss how recent technological achievement have enlarged our understanding in the function and mode of action of plant RNA binding proteins. Small and colleague also take a look at RNA binding proteins with respect of RNA processing and maturation in organelles, mostly in chloroplast and mitochondria. Then Ezequiel Patricio, a good friend of mine, do an intron-centric guide to alternative splicing, where he discussed the fundamental aspect of splicing and alternative splicing, but focusing mostly in introns. Palos and colleague provide a perspective on long non-coding RNA, discussing key discoveries in the last 25 years, how this molecule challenges most of the stuff that we know about central dogma, and discuss new molecular and computational approaches to analysis of these molecules. Then Cho and Mosher review small RNA-mediated DNA methylation during plant reproduction, focusing on biosynthesis, transport, and function during reproduction. Proal and colleague provided an overview of covalent nucleotide modification within plant mRNAs, the method to detection and analysis, and the outstanding question that remain to be solved. Then, as I mentioned, we have 7 research articles in a breakthrough report. Hudzik will report trans-species microRNAs in the parasitic plant Cuscuta campestris, showing that they have a UC-like promoter. I'm not going to go into this paper because he will present it today. Then Guo and colleague in large-scale biology will present a study from the translational landscape of wheat during grain development. And then another 5 articles, 2 of them that will be featured today, discuss the novel classes of small RNA upregulated by nutrient deprivation in Chlamydomonas. It's one of the articles that we will feature today, as well as the structural insight into the activation of organelles CTU RNA editing in Arabidopsis. The other 3 articles will be featured in the next webinar and discuss the functional dependency of subunits of Arabidopsis N6A methyltransferases, the discoveries of the requirement of D and E1 endonucleases for turnover of mRNA, and the study of synergistic action of spliceosome components during post-transcriptional gene silencing. Today webinar will be moderated by our plant cell assistant feature editor, Sara Lopez-Gomollon, who will introduce each of the speakers and handle the question. Sara completed her PhD in the University of Zaragoza in Spain. Then she carried out the postdoc work on microRNAs and other small RNAs in Thomas Dalmai at the University of East Anglia, and also with the legendary C. David Bolcom at the University of Cambridge. She is now a Broadbank Senior Research Fellow at the University of Cambridge, where she's working on discovering the novel mechanism regulating gene expression by regulatory small RNAs associated with transposons. Without much to say, we shall pass the screen and microphone to virtual microphone to Sara, so she can introduce the speaker. Thank you.
Thank you very much, Pablo, for the nice introduction, and welcome. Also, as Pablo was saying, we have an amazing program today, so these three papers will be covered, and we will start by Sachiko. So Sachiko Toma-Fukai is an associate professor in structural biology at the Nara Institute of Science and Technology in Japan. So she did her PhD in macromolecular science at Osaka University and was an assistant professor at the University of Tokyo before moving to Nara as an associate professor in 2018. So Sachiko is interested in the molecular mechanisms of post-translational or post-transcriptional modifications and how we can, especially about how we can relate those to therapeutic strategies and how to combat disease from the viewpoint of structural biology. So her talk was recorded, but she will be here for questions and answers that you can please write them down in the chat below. And today she will present her recent work that is entitled Structural Insights into the Activation of an Arabidopsis organelle C2U RNA Editing Enzyme by Active Site Complementation. So thank you, Sachiko.
My name is Sachiko Toma-Fukai from the Nara Institute of Science and Technology. First, I would like to thank the member for preparing this webinar and giving me this great opportunity. I am pleased to be here and to have a talk about the recent structural study. The title of today's webinar is Structural Insight into the Activation of an Arabidopsis organelle C2U RNA Editing Enzyme by Active Site Complementation. In plant organelles, sequence-specific C2U RNA editing occurs in all plant plants except for Machantia. Cysteine deaminase catalyzes the conversion reaction from cysteine to uridine. RNA editing is a post-transcription process. This modification changes the sequence of RNA without affecting the genome DNA. The C2U conversion can create the start codon, change the amino acid, or create the stop codon in mitochondria and chloroplast genes. In angiosperms, there are more than 400 sites in mitochondria and 30 to 40 sites in chloroplast. As you can see in these photos, some mutation in the RNA editing factors lead to severe phenotypes, also suggesting that RNA editing is essential for plant organelles. Up to now, over 100 pentatricopeptide repeat PPR proteins have been identified as C2U editing factor in plant organelles. In this slide, I show the domain diagram of OTP86 here. This is an editing factor in plastid and one of the typical PPR type RNA editing factors. The N-terminal PPR domain recognizes single-strand RNA, while the DYW domain catalyzes a C2U editing reaction. E1 and E2 have PPR-like motifs, but so far the function is unclear. Half of the PPR-type RNA editing factors harbor both PPR and DYW domains. Amino acid sequence analysis indicated that the DYW domain would be a novel deaminase hold. Interestingly, another half of PPL type RNA editing factor in Arabidopsis lacks the DYW domain. Genetic analysis suggested that such a C-terminal truncated PPL proteins needs a DYW1-like short protein as a partner, which consists of no or only a few PPL motifs and a partial DYW domain. In this slide, I show the domain architecture of the protein CRR4, which is required for converting an editing site in NDHD in plastid. CRR4 need protein DYW1 to exert RNA editing activity. Interestingly, the DYW1 protein lacks the PG box region of the DYW domain, while CRR4 has its instead. The structural mechanism underlying the activation of the DYW1-like deaminase has not been revealed, so we performed the structural study focused on Arabidopsis DYW1. We successfully determined the crystal structure of the DYW1 domain, DYW domain of DYW1 at 1.8 angstrom resolution. The structure shows that DYW1 hold into the single domain in the C-terminal region. On the right, I show the crystal structure of already determined DYW domain of OTP86. As you can see, the whole architecture looks different from each other. The N-terminal region corresponding to the gating domain that was proposed to be involved in catalysis regulation in OTP86 is significantly different. The DYW1 consists of alpha helices, while OTP86 has 2 beta strands. The function of this region of DYW1 is so far unclear, so we named it the DYW deaminase insertion region, DDI region. Next, I will show the structural detail of DYW1. The whole structure of the DYW domain of DYW1 is divided into 2 regions, the DDI and the deaminase domain. DYW1 has 2 functional zinc binding sites. One is zinc 1, located at the active site. The active site structure is highly conserved in deaminase family, and the arrangement of the residues relating to the deaminase indicated that this structure is active home. Another zinc called zinc 2 is located near the conserved DYW motif. This C-terminal conserved DYW motif, including zinc 2, SIM2 work for structural integrity. A striking structural difference was found by comparison with the crystal structure of OTP86. DYW1 lacks the beta sheet corresponding to the PG box in the OTP86 structure. I'm showing the PG box sequence here. DYW1 doesn't have a PG box. Instead, CRR4 has a PG box. So I show the CRR4 sequence here. From the OTP86 structure, we found that the residues involving interaction with the deaminase domain are highly conserved. As you can see, the CRR protein has the residues. We built a domain model structure of the DYW domain of DYW1 and PG box structure based on the OTP86 structure. The model structure shows that the unique residues of CRR4 indicated here are exposed to solvents outside of the complex molecule. The PG box of CRR4 could form a complex with DYW1 in a similar manner. We carefully compared the structure of the active site of deaminase structure deposited in PDB and found that all deaminase has conserved 5 beta strands, and the beta strands corresponding to the PG box make the substrate binding pockets, shown as the blue sheet and violet loops. The Eugene docking model of OTP86 structure shows that OTP86 lack a part of a catalytic pocket. The crystal structure of OTP86 does not include an E2 loop. The result indicated that a part of E2, in addition to PG box, is also part of the active pocket. Violet loops recognize the sugar moiety of Eugene. Part of the violet loop is the E2 region. DYW1 does not have an E2 and PG box in its molecule. Therefore, DYW1 lacks a large part of the catalytic pocket shown here. This result indicated that DYW1 only would not provide a suitable catalytic pocket to accommodate substrate. To corroborate this hypothesis, we conducted an in vivo RNA editing assay. We made a chimera protein whose focus on PG box region. Chimera protein harbor PPR E1 and E2 domain of PPR56, which is PPR protein having a C-terminal DYW domain. As you can see, the chimera protein lacking PG box lost the editing activity. The protein harboring the first beta strands recovers its activity to 50%. The editing activity of the protein having PG box of CRR4 greatly enhanced to about 90%. The result indicated that the PG box is essential for the C2U RNA editing activity. Finally, we propose the activation mechanism of DYW1. CRR4 complements the lacking PG box of DYW1 by forming a complex, and then DYW1 is activated. This activation model would be the common mechanism of DYW1-like protein. I would like to summarize our study. We determined the crystal structure of the deaminase domain of DYW1. Structure evaluation and in vivo RNA editing assays reveals that DYW1 is activated by forming a complex with CRR4. We demonstrated the common activation mechanism of the DYW1-like proteins by molecular complementation. Please visit our paper published recently. I would like to thank all people shown here. All structure study had done in Shimizu lab at the University of Tokyo. My present affiliation is the Nara Institute of Science and Technology. In vivo RNA editing assays were performed in Takenaka lab at Kyoto University. We collected diffraction data at KKPF Tsukuba, Japan. Thank you very much for your kind attention.
Thank you very much, Sachiko. So please, if you have any questions, you can use the questions and answers or the chat below. But while the questions are coming, I have a question for you. So do you think that there is potential for this domain to be manipulated in a way that could be used as an on-demand RNA modification genetic tool?
I think it is possible, but to use the to develop that tool, I think the more structured study is required. So because I just determined the DYW domain, structure only, but we have to determine the include the PPL and more structural detail of the activation mechanism. That's this molecule, I think that very potential molecule to use something the RNA modification manipulation. Thank you.
So actually, I can ask you something else. So what do you think? So we have some PPR proteins that have this domain that is part of one unique transcript. And then we have others that have the full functional protein divided into transcripts in a way with these two different domains, and the one that you identify the structure in this paper. So why do you think that could be I'm thinking in evolution, why do you think that these proteins have opted for these two different structures for these genes? So do you think that could be related to regulation or?
That is a really good question, but I don't have the answer now, so sorry.
No, thank you. So we have some questions in here. So Daniela Lips is asking, what determines sites are edited? So if it's a sequence specificity, and if can one predict if a site will be edited or not from the sequence?
PPL region determine the binding site of RNA. So, but divide it has not been revealed yet, but divider probably don't have a specificity of the RNA and PPL region. So in this complex, the CCR4 determines the region of the binding site. So PPL domain and previous structure study show that specificity to the sequence binding specificity for PPL.
Mm-hmm.
So is can I?
Yeah, so one domain is giving the specificity for the sequence to edit, and the other domain is acting on that. Okay, thanks. There's just another question. Is asking if you are aware of any study regarding C2U editing on rice.
I don't know. It's probably the not yet. I think.
Oh, there is another question over here that would be probably the last one. So it's asking, what is the significant functions of this domain in agriculture crops?
Additional to probably no. It is so this question means that other RNA is required for to exert the modification activity. Probably I just do the in vivo RNA assay. Probably I think that we could not experiment in the purified molecule, for example, the RNA, purified RNA and purified protein, but I don't know well, but I think that it's not need the additional RNA.
Mm-hmm. Okay, thank you. So we have just one last question, and maybe we need to move on. So Valdir Moreira said, very nice talk, and I agree. This person would like to know if this protein required additional RNA to modify for C2U modification. So it's asking if this system is related to something like CRISPR-Cas9 that use a guide RNA. So this person is asking if it's needed like an additional RNA for that.
Well, I think that I think that in this reaction, probably the additional RNA is not required, not needed, I think.
Right, because the protein is the one that is bringing the protein to that sequence, right? The domain that you were mentioning. Okay, thank you. very much, Sachiko, for this interesting talk. So we will now move on to the next. Oh, there is one last.
Yeah.
Yes, exactly. Thank you very much.
Thank you very much.
I was just catching up with the questions.
Thank you very
so
much.
we will. Thank you very much, Sachiko.
Thank you for having me.
So we will now move on to the next speaker, that is Yingshan. So he is an associate investigator at BGI Research in Shenzhen, China, where he is interested in expanding the genetic code of eukaryotes using Saccharomyces cerevisiae as a model. So he did his PhD in genetics and also a master in computer science at the University of Nebraska, Lincoln, after completing a bachelor in biotechnology at Huasong Agricultural University in China. So Yingshan is interested in understanding the biogenesis and molecular functions of non-coding RNAs. And today he's going to present a chapter of his PhD, where he studied small RNAs in Chlamydomonas reindariety and linked those small RNAs to stress response. So the title of his talk would be Small RNAs over 26 nucleotides in length associate with AGO1 and are upregulated by nutrient deprivation in the algae Chlamydomonas. So thank you, Yingshan, and the stage is yours.
Can you hear me?
All good,
Hey.
Yingshan.
Hey, can you hear me?
Yes, we can.
Okay, thank you very much. All right, hi everyone. I'm Yingshan Li. Thank you so much for having me today and for joining us. Today I will be presenting my PhD research exploring a new species of small RNAs, longer than 26 nucleotide and are associated with Argonaut-1, and they are upregulated by nutrient deprivation in the algae Chlamydomonas. The first question is, why are small RNAs important? Small RNA can mediate RNA interference, a process that commonly leads to gene silencing through degradation, RNA degradation, or translational repression of complementary messenger RNAs. The core RNAi machinery and mechanism in eukaryotes have been well established. First, the microRNA genes are transcribed into hairpin structures. And this hairpin structure, as well as the double-stranded RNAs, can be recognized and processed by dicer proteins into small RNA duplex, which are then loaded into RNA-induced silencing complex. And this complex will then be guided by the small RNA to target complementary transcripts for either degradation or translational repression. And the core effector of the silencing complex is the Argonaut protein. So we can see three essential components of RNA interference: the small RNAs, dicer protein, and Argonauts. And functional-wise, RNAi play important roles in gene regulation, antiviruses, and transposable elements silencing, and so on. And our lab has been using the unicellular eukaryotic microalga Chlamydomonas reindariety as the model. Chlammy was the first unicellular eukaryote where the microRNAs were discovered. It has a relatively simple but complete set of RNAi machinery with 3 Argonaut proteins and 3 dicer proteins, as well as a variety of small RNAs. And our first phylogenetic analysis showed that Chlammy AGO1 and AGO2-3 form into 2 different clades. However, AGO1 seemed to have been lost in many of the lineages in Chlorophyceae, such as Volvox. And our lab, as well as other groups, have established before that Argonaut-3 in Chlammy is the major Argonaut that recruit canonical microRNAs to regulate endogenous genes. And Argonaut-2 is a very recent duplicate of Argonaut-3. They have more than 90% of similarities in protein sequences. and is so assumed to have redundant rows with Argonaut 3. But Argonaut 1 seems to be much more divergent than the other two paralogs, with no known functions. So Argonaut 1 in Clemmy is the focus of my study. And it was shown before that in Clemmy, microRNAs are mostly 21 nucleotide, but surprisingly, we went to sequence the total small RNAs in Clemmy, and we found that besides the 21 nucleotide microRNAs, there is also another type of small RNA distinctively longer than 26 nucleotides, which has never been seen before in plants. And this long small RNA, we will call them long small RNAs. And they comprise over 40% of the total small RNAs. We know that microRNAs are produced and precisely loaded into one of the Argonaut 3. So we wonder if the other type of small RNA also load into one of the Argonauts. So we immunoprecipitated Argonaut 1 and saw that these long small RNAs actually precisely associated with Argonaut 1 instead of Argonaut 3. So we see here is a very elegant scene that Clemmy produced two different types of small RNAs and are differentially loaded into two different types of Argonauts and presumably play different roles. And this new type of small RNAs can be readily detectable by northern blot analysis. And it has not been seen in other plants, either in other algae, moss, gymnosperms, or angiosperms. To further corroborate our conclusion that these long small RNAs are loaded into Argonaut 1, we went to knock out Argo1 with CRISPR-Cas9 and obtained two independent mutants. And we could see a reduced level of long small RNAs in Argo1 mutants, indicating that they are actually loaded into Argonaut 1, although some of the long small RNAs are not completely abolished and may persist by alternative association with intact Argonaut 2 and 3 proteins. And our further genomic analysis shows that the long small RNAs are produced from genomic clusters. Unlike microRNAs, we know that they come from all over the place in the genome. And large amounts of long small RNAs are mapped into only several hotspot clusters. And these clusters, if we zoom it in, we can see that they are uniform in length of around 70 to 80 nucleotide. And very interestingly, we found that this long small RNAs precursors can fold into hairpin secondary structure, which have two loops and two stems. And the cleavage sites are very conserved in one end with stem structure, and but more of randomness in the other with loop structure corresponding to here. And we can also detect the precursors by northern blot here at about 70 to 80 nucleotide. So here it comes to the biogenesis of these small RNAs. We knew before that the canonical microRNAs are processed by dicer3 mutant, by dicer3 protein. We wonder if these long small RNAs are also dependent on dicer proteins. So we compared the total small RNAs in wild type and dicer3 knockout mutants. Well, we could see that the 21 nucleotide microRNAs are mostly abolished in dicer3 mutant. The long small RNAs are not affected by knocking out dicer3, suggesting that these long small RNAs are independent of dicer3. And this agrees with our wet experiments. We further tested the abundance of long small RNAs in the knockdown mutants of the other dicer1, and showed that the long small RNAs are even more abundant. We can see that they're even more abundant than in wild type CC124, suggesting that they are independent of dicer1 too. As I mentioned earlier, Clammy encodes 3 dicers. So we went to knock out the last dicer protein, the dicer2, with CRISPR-Cas9, and found that dicer2 is not responsible for the long small RNA biogenesis either. So far, I think we have demonstrated that the long small new type of small RNAs are independent of neither of the 3 dicer proteins, although we still don't know which ribonuclease is actually involved. Other than the biogenesis, we were also curious about the evolution of this long small RNAs. How do they come from? Where do they come from? We found that these clusters are actually conserved in related species, which is unusual, because usually small RNAs, they're too short, unlike the exon regions mostly conserved. Small RNAs like microRNAs are usually not conserved from the perspective of sequence. Very interestingly, we see that the number of clusters have been expanding throughout the evolution of in Chlorophyceae clade. We can see that there are 19 homologous clusters in Chlamydomonas diberiana, and we have 87 clusters in Chlamydia scloesri, and 352 in Chlamydia incerta, and 324 in Chlamydia reinhardti. This suggests that these long small RNAs originate from repetitive elements and originated after the divergence of Chlorophyceae, because we don't see any homologous heat in Volvox or Chlorella. And this happened about 800 million years ago right here. And so far, I think our data has demonstrated that the long small RNAs are under high evolutionary constraints, indicating conserved functions. But what exactly are they? Now let's go back to the gene that contain multiple long small RNA clusters, this intron of this gene. And this gene encodes Fra2, a ball-like protein, which plays a role in regulation of iron metabolism genes through sensing ferrum sulfur cluster synthesis. Very interestingly, this long small RNA produced from its own intron can potentially regulate itself by binding to its 3' UTR, because these two sequences are complementary perfectly to each other. And very, very amazingly, we can see that even the targeting site is conserved in other species. In the meantime, we show that these long small RNAs are induced under some nutritional stresses, such as sulfur and nitrogen starvation. And to verify that this long small RNAs can regulate the expression of that gene, we analyzed the transcriptome data of Chlamydia under different stresses. We found that the expression of this gene actually decreases under this same stresses in accordance with the upregulation of this long small RNAs under the same stresses. And lastly, our phenotypic analysis showed that the loss of AGO1 has also yielded a phenotype that becomes chlorotic faster than the wild type under sulfur starvation. So to summarize, our data demonstrate that Chlamydia produce both short microRNAs and long microRNAs, which are loaded into different argonauts. And secondly, the long small RNAs are independent of dicers, whereas their precursors have conserved a secondary structure. And thirdly, the long small RNA clusters have been derived from moderately repetitive elements, although we don't know if they are transposable elements, but they have been expanding in the core Chlamydomonas. And lastly, the long small RNAs can be significantly upregulated under nutritional deprivations, and they can regulate the expression of complementary transcripts under these conditions. And with that, I'd like to thank my PhD advisors, Dr. Herberto Cerruti and Dr. Esko Moriyama, as well as my PhD committee for all of their guidance and help. I'm happy to answer any of your questions. Thank you very much.
Thank you very much, Yingshan. Amazing talk. So we have some questions in here. So we have one that was actually asked by Pablo Manavella and Xuemei, that it's related to the fact that the long small RNAs seems to be, the long small RNA seems to be independent on dicer 1, dicer 2, and dicer 2. So they are asking if you check whether all three are redundant in case, if you have a triple mutant, so in case that any of them could generate them, and it was actually dependent on dicer, but just any dicer.
Yeah, that's a very good question. Actually, when this paper was being reviewed, our reviewers asked the same question. They, we had supplementary data showing that only dicer 3 has relatively high expression level. Dicer 1 and 2 usually are have very, very low expression level, and when we knock out dicer 3, we don't see a increase of dicer 1 or 2. So we figure that there is or very low amount of compensatory effect of this 3 dicers. Although I agree that if we can actually have a triple mutants of dicers, that would be even better. But unfortunately, we didn't have. That's a very good question.
That's for the next paper then to confirm that. We have another question. We have lots of questions, so I would try to answer as many as possible. That if I would try to ask as many as possible. If we run out of time, I'm sure that Yingshan would be happy to answer them on the chat. So Marco in Carbone is asking if you have any clue about what RNA polymerase makes these long small RNAs, whether or not dicer is required.
Yeah, that's a good question. Usually the microRNAs genes are synthesized by PO2, but this we didn't test which polymerase is involved, but it seems like it's not very long. It's only about 70 to 80 nucleotide, probably PO2, but actually we don't know.
So Dr. Ravindra Dubey is asking if these if the function of long small RNAs is known. That I think you answered partially that during your question. Basically, if they have any kind of temporal regulation.
Oh, say it again. I'm sorry, I didn't get that.
This person is asking about temporal regulation of these long small RNAs. So you showed that were related to stress. I don't know if you did any temporal analysis of these small RNAs, like with development or different tissues.
Yeah, they are as I just showed. Usually they are at very low amount of expression level, but when they are under stress, with no nitrogen, no sulfur, they're extensively upregulated, and seems like it's in accordance with the downregulation of its target genes. But under normal conditions, we're not sure, but we postulate that this small RNAs mainly play a role in the tolerance to different nutritional stress. Well, one interesting thing to point out is that I mean, in laboratory conditions, we grow cells, we give them a lot of nutrition, but actually in the wild, there is no there is no nitrogen, there is no sulfur, and we say stress in lab is the actual environment for them in nature.
Indeed, yeah. So you think that probably in nature these small RNAs could be more widespread than what we can find
That's
in the
what we
lab.
think. Yes.
And I'm going to ask just the last question. There are a few that I welcome you to check on the chat so you can reply to them. So the last one, it's from Pablo as well. So he asked, lone small RNAs appeared reduced in AGO1 mutants. Any idea why? Is AGO1 protecting them from degradation?
Yeah, probably. So if this, I mean, usually they are loaded into Argonauts, and the loaded small RNAs on Argonaut are relatively more stable. But if Argonaut 1 is knocked out, some of them will turn to the other two Argonauts, but the rest will probably be degraded.
Okay, okay, thank you very much, Yingshan. And as I said, like, please go ahead and answer, type the answers for the questions that are in the chat that we didn't have time to cover in here. I apologize in advance for not being able to cover all, but well, it was, as the first one was a very interesting talk. Thank you, Yingshan. And now I will introduce to our, thank you. Now I will introduce to our third speaker today. That is Collin Hudzik. So he completed his bachelor and master degrees at Bridgewater State University, Massachusetts, and he did his PhD in biology at Penn State. So his PhD focused on microRNAs in the parasitic plant dodder, Cuscuta campestris, and these microRNAs have trans-species activity and can target mRNAs in the host plants, like for example, tomato. So today, Collin will present some of his work that was included as part of his PhD, and that he has summarized in the talk that he's going to present today, entitled Trans-Species microRNAs loci in the parasitic plant Cuscuta campestris have a U6-like snRNA promoter. So please, Collin, stage is yours. We are looking forward to your talk.
Awesome. Thank you so much. All right. Can everyone see my presentation up?
Yes, we can see your presentation.
So I'm just going to give a brief outline to how this is going to go. I'm just going to give a background on microRNAs in general and talk a little bit about the parasitic plant that I work on. It's a really interesting system, and not everybody knows about it. So I'll just talk the brief, interesting parts of Cuscuta, and then I'll kind of break down my paper into really the three main takeaways that I want you to have at this presentation. I'm not going to be able to include everything that I want to from the paper, as we included a lot, but I'm hoping that this will kind of kickstart everybody into wanting to read more about the paper. I know we just had kind of a introduction into the biogenesis of microRNAs in plants. I'm just going to go over it one more time. So microRNAs are transcribed by RNA polymerase II. They create this single-stranded molecule called the primary microRNA that kind of has an extra tail and this hairpin structure that gets rapidly processed by a group of proteins that go through kind of two big processing events where it removes the arms, it becomes a pre-microRNA, and then that hairpin at the top gets removed as well, and kind of left with this microRNA star duplex. One of those strands, the microRNA star, gets rapidly degraded, and the mature microRNA gets bound to an Argonaut protein. This Argonaut protein will then leave the nucleus where it can go moving around within the cytoplasm to try to identify any sequences complementarity, and it'll end up degrading. The big takeaways that I want to state here is that there's really no promoter elements in microRNAs other than like chatterbox, and all plant microRNAs are transcribed by RNA polymerase II. The parasitic plant that I work on is in the genus Cascuda. It's a genus of stem holoparasites. And what I mean by that is Cascuda lives on the stems of its host plants. And the holoparasite means that it cannot complete its life cycle without being on a plant the entire time. It lacks any kinds of roots and leaves. It doesn't produce enough chlorophyll to sustain its own life, so it ends up kind of having this yellowish color. It's known to be a generalist parasite, so it can basically attach to any dicot plant and very rarely some monocots. It uses this organ called the haustorium. The haustorium will invade into the host tissues where it'll fuse with the host vasculature, both the xylem and the phloem. That's how it receives its nutrients during its life. So this haustorium is known to facilitate bidirectional movement of all kinds of molecules, including proteins and RNAs. So if we were to kind of just look at a cartoon of a Cascuda growing on an Arabidopsis of inflorescence, this area is what we call the interface. So it's the area where the parasite and host are directly interacting. Cross section of this and kind of look in, you can see that the parasite is beginning to invade into the host tissue and it's growing towards the vasculature within that stem. And after a certain amount of time, these cells which are invading into the host tissue will interact with either xylem or phloem, and that is when they de-differentiate and redifferentiate into either xylic or phloic hyphae, what they're called. And that kind of allows this bridging for the parasite to receive nutrients from its host. Haustorium organogenesis occurs in three main phases. I'm not going to dive too deep into them, but I just want you to know the adhesive phase is this initial contact between the parasite and the host. Cortical cells within the parasite begin to differentiate to what will end up becoming a haustorium. You break into the intrusive phase once these searching hyphae extend out of their own tissues and begin to invade into the host tissues. They're going to be looking for both the xylem and phloem, and you've reached the conductive phase once the parasite is actively receiving nutrients from the host. This is typically denoted by a xylem bridge, just because it's really easy to identify xylem bridge as compared to other kinds of cells to get that really nice striation pattern. So we do have an understanding of how long haustorium organogenesis takes. We don't really know about the individual phases. Our lab previously had identified that at the interface between the parasite and the host, so again, directly where they're touching, Cascuda is producing a lot of microRNAs. We ended up terming these as interface-induced microRNAs, as we're not able to see them in any other parts of the parasitic plant unless it's actively creating haustoria and touching the host plant. When looking at these interface-induced microRNAs, we had found that some of them have trans-species activity. What I mean by that is that these microRNAs are moving from the parasite into the host and regulating host gene expression, often involved in like phloem proteins, defense-related proteins, auxin sensing, and stem cell division transcripts. And if we look at this graph here, it's from our publication in 2018. Red box plots are where the parasite and host are interacting. So that's interface tissue and then just a control stem. You can see in all cases that we had looked at, when the parasite is growing on the host, you're getting lower accumulation of these transcripts that are targeted by these interface-induced microRNAs. So we have identified this population of microRNAs in Cascuda at the interface. We term them these interface-induced microRNAs, and we know that some of them can move from the host from the parasite into the host and regulate gene expression. We have some evidence that these may be used to increase the parasite's fitness during infection. But what I really sought out to investigate, what I'll talk about in this presentation, is when are these interface-induced microRNAs detectable during infection? and how are they so tightly regulated to only be expressed within the interface? So what kind of cis-regulatory elements or promoters could we identify upstream of these loci? So I'll jump right into my time course. All of our previous work that we had done to identify these interface-induced microRNAs were always done about 10 days after the parasite had wrapped itself around the host. So we had really no idea when these turn on. And identifying this could really be helpful for having us understand, are these interface-induced microRNAs really important to the beginning stages of parasite infection? So I ended up creating this time course where I would initially place Cascuda around the stem of either Arabidopsis or tomato. I use both hosts. Once Cascuda wraps around its host, I consider that day 0 of my time course. And every 24 hours after that was just another day within the time course, up to 14 days. From these samples, I would either use them for small RNA library preparation, or I would take the interfaces and do vibrotome sectioning, so I can correlate my small RNA-seq data with trying to understand which phases of Haustorium organogenesis can we identify the microRNAs in, as well as secondary siRNA accumulation. So this is kind of our big figure within this paper, where we have tied in the vibrotome sectioning in with our small RNA sequencing data. So the first 3 photos are Cascuda growing on Arabidopsis, and then the next 3 photos are Cascuda growing on tomato, where we had basically taken the best photo for which phase of Haustorium organogenesis are we on, and tied that kind of in this RNA sequencing data here. The big thing is that Haustorium organogenesis really is progressing at the same rate, despite growing on 2 very different hosts, where we have kind of a tomato hypocaudal, which is essentially a baby, and then we have a Arabidopsis inflorescent, which is kind of at the end stage of its life as well. And if we look down here, this is all of our interface-induced microRNA loci plotted on the box plot per day. And you can see very abruptly in both Arabidopsis and tomato, they start a lot earlier than we had initially predicted of when we'd be able to detect them. And if we look, you can see that it starts at about day 2 for Arabidopsis and between day 1 and day 2 for tomato, and kind of tying that in with Haustorium organogenesis. This is within the adhesive phase of Haustorium organogenesis. So this is before the parasite has even invaded into the host tissues. You get this ramping startup of these interface-induced microRNAs accumulating and preparing for themselves to be sent over into the host tissues. So I'm going to talk about this element now that we had discovered upstream of the interface-induced microRNAs. So we found it really surprising that all the interface-induced microRNAs turned on so early in infection, and they all turn on at the exact same time. So we ended up looking upstream as well as downstream of where we were predicting the coordinates of all these interface-induced microRNAs. So we looked at the 5' lower DCL cut site and the 3' lower DCL cut site, and kind of just went up and down stream 200 nucleotides to look for any kind of terminator signals, as well as dysregulatory elements or promoter elements. And in 93% of the 156 interface-induced microRNAs we have locations for, we found this really strongly upregulated 10 nucleotide motif, TCCC, ACA, CCG. We did a little bit of research to try to find if anyone else had ever reported this motif before, and it was actually identified back in the 90s as the exact same cis-regulatory element that's used to drive small nuclear RNA expression in plants as well. This is known to be a really strong constitutive promoter, and it hasn't been identified anywhere else except within snRNAs. So we wanted to see how important is this upstream sequence element for accumulation of these interface-induced microRNAs. It's really tough to do any kind of genetics within Cascuda. So we ended up using agroinfiltration, where we created two different plasmids. Essentially, one was the wild type version of two of the interface-induced microRNA loci, where you have this upstream sequence element, the wild type sequence of the interface-induced microRNA, as well as downstream of this as well. And then a second version where we essentially just scrambled the sequence of the upstream sequence element, perform agroinfiltration, and we did QRT-PCR. And you can see in both cases of microRNAs that we had tested, accumulation is only detectable when we have this upstream sequence element acting as a cis-regulatory element. So we think that this upstream sequence element is definitely important for accumulating these interface-induced microRNAs. A little bit more background into snRNAs. snRNAs are really unique as the distance between the upstream sequence element and the transcriptional start site actually determines which RNA polymerase will end up transcribing these loci. So if you have a distance of about 66 to 67 nucleotides, it's going to be transcribed by RNA polymerase 2. And if you have a shorter distance, 54 to 55, it's going to end up being transcribed by RNA polymerase 3. So knowing this, we wanted to look at the primary transcripts of these interface-induced microRNAs. And we also wanted to be able to test what their 5' ends looked like. So were they either triphosphorylated, which is a sign of RNA polymerase 3 transcription. And then we also wanted to see if they had a cap, which is due to POL2 transcription. So we devised this RNA sequencing method to capture full-length primary transcripts, and then allow us to do these treatments on them, which will discriminate if they're transcribed by RNA polymerase 2 or RNA polymerase 3. So we have some kind of known POL2 and POL3 transcripts, which we used to help us determine what the interface-induced microRNA loci would be. So if you have a cap, which means you're transcribed by POL2, in both of our treatments with this VCP and RPPH, you'll accumulate even levels in both of the treatments. And if you are transcribed by RNA polymerase 3, you'll end up only accumulating in this RPPH treatment. So this is just known caps RNAs. This is pre-tRNA transcripts, which are POL3 transcribed to have that 5' triphosphate. And if we look at the interface-induced microRNAs, you can see that they accumulate essentially the exact same way that other known POL3 transcripts accumulate. Also letting us know that they don't have, in any cases, a 5' cap. So we know that this kind of fits as well, that they have this kind of shorter spacing that allows for POL3 transcription. And when we look at their 5' ends, we see that they're not like POL2 transcripts. We also decided to look downstream of these interface-induced microRNA loci. So POL3 transcripts only need a string of about 4 Ts to be a strong terminator for POL3 transcripts. So at first we did plot how far away this distance between the upstream sequence element and the transcriptional start site was for all of these interface-induced microRNA. And in most cases, they were about 55 nucleotides away, distance between the use and the transcriptional start site. And nearly all of them ended at poly-T regions directly downstream of the microRNA loci. So I'm going to give takeaways and kind of talk about the points I want everyone to be able to take home. So interface-induced microRNAs are accumulating really early during infection. So once Cascuda wraps around its host and it begins to form these hostorium, before it's even broken through into the host plant, it's already generating these interface-induced microRNAs at about day 2, so about 48 hours after it's wrapped around a host. We identified this upstream sequence element to to be within 93% of what we're calling interface-induced microRNAs. We found that it's vital to their accumulation when doing agroinfiltration in the heterologous system within Nicotiana. And all of our evidence that we have currently is suggesting that these are transcribed by RNA polymerase III. They don't have a 5' cap. They all terminate at poly T sites. And the big thing really is, who cares? What is the big significance that these are transcribed by RNA polymerase III? So I think someone had actually said it before, but all plant microRNAs are known to be transcribed by RNA polymerase III. So there's never been a small RNA, a microRNA that has been categorized to be transcribed by pol3. And there are also no promoters outside of the Tata box, which which any microRNAs have in plants, especially ones that are pretty divergent. So we have, a list of 156 interface-induced microRNAs, and we've broken them down into about 103 families, and all of these families contain this 10 nucleotide motif acting as the cis regulatory element. So we think that maybe transcription by RNA polymerase III might mark these interface-induced microRNAs for special export. So we don't have any evidence that any of these interface-induced microRNAs are actually targeting transcripts within parasite tissues. We've never been able to see any kind of splicing events occur within parasite tissues. Only within host tissues do we see this down regulation of host transcripts. So we think that possibly transcribing by RNA polymerase III allows these microRNAs either as a duplex or just as their mature strand to avoid being loaded by AGO1 and being packaged or some kind of mechanism to be sent directly into host tissues to avoid their own self-targeting events. We're kind of working on that right now, and we're really excited to look at these data as they're coming in, but I don't have any evidence yet for completely avoiding AGO targeting yet. I do want to thank everyone in my lab. A special thanks to my advisor, Dr. Axtell. I wanted to include my funding, and then also a shameless plug to include my email. I just finished defending a little over a week ago, so I'm officially on the job market. So if anyone wants to talk more about this project, this presentation, or have any jobs that they think I would fit well in, please don't hesitate to reach out to me. With that, I'll take a question.
Thank you very much, Collin. Very interesting talk as well. So then from your talk, I understand that these interface-induced microRNAs in Cuscuta have this pol3 promoter, while the rest of microRNAs have a traditional pol2 promoter, right? Okay,
yeah,
so the question.
So the upstream sequence element isn't necessarily only a pol3 promoter. It can be pol2 or pol3 transcribed, but it's the distance that determines which one will end up transcribing that region. And the interface-induced microRNAs do have that pol3 distance.
So you say that non-interface microRNAs may do have that motif?
No, so only snRNAs have that motif.
Okay.
And they also have that discrimination between being transcribed by pol2 or pol3 depending on distance.
Oh, okay, I see. So one of the questions from Pablo was that from an evolutionary point of view, do you have any clue how these microRNA promoters specialized?
So that is something that is talked about in our lab a lot. We don't really have any idea as to exactly how all of these 156 interface-induced microRNAs could have grabbed this snRNA promoter. It could be due to whole genome duplications as well as tandem duplications. We do see in a lot of cases that these interface-induced microRNAs are kind of set right after each other within the genome. But it's something that our lab is definitely interested in looking into and trying to figure out how this could happen.
I have a question related actually to this. is that this type of sequences will be part of a constitutive promoter, right? So you would expect a constitutive expression based on those motifs. So
Yes.
do you expect any other regulatory motif that could be present in these microRNAs or any additional regulatory layer related
so
to
that's
infection?
that's something that we're definitely looking into as well. So yeah, these this upstream sequence element, that sequence is known to be really, really strong constitutive promoter. People use it to drive small RNA, small guide RNA expression within CRISPR experiments. So, how is it that this constitutive promoter only expresses within the interface? We're trying to figure out some good experiments to kind of clean that up and see if we can find anything else that's maybe binding or inhibiting this region from being transcribed. But it's definitely perplexing.
Thanks. We just finished. I'm going to combine two questions in here. So first, Rodrigo Machado is asking if Cuscuta invade the phloem and xylem, and if it has a preference for a specific tissue. And then we have another person asking if these small RNAs move to the host plant naked or associated to any kind of protein. Do you have information about that?
So the first question was, does the parasite prefer either xylem or phloem? I think that's so it ends up creating both xylic and phloic hyphae. So there are multiple areas where the parasite will attach, all within the stems, or basically wherever it's touching. And in each one of those regions, it'll connect to the xylem and the phloem. We haven't really counted to see, how many connections per hostorium does it actually make, but it's really difficult to kind of see a true phloem connection. Usually people just look at that xylem bridge just because it's really easy to determine where it is. And then the second question was
if the small RNAs were naked or associated to
RNA. So we don't have any evidence yet for either case of that. It very well could be that maybe there's some kind of protein that is binding within parasite tissue and then being moved into the host where that microRNA ends up getting released or something. But that's something that we're currently thinking about looking into more as well.
Okay, so thank you very much, Collin, and the rest of speakers. It was great. If some questions if you have any remaining question, please write it down in the question and answer box, and I'm sure that our speakers will be happy to answer them. And with that, I will just pass the microphone back to Pablo for a wrap up of the webinar.
Thanks, Sara, and thanks to all the speakers. It was an amazing session. So after that, I just want to thank all of you to be here. And I want to remind you that this is just the first part of the webinar. There's going to be a second edition. It's going to be on 28th of June. It's going to be hosted by Becky Mosher and moderated by Nora Flynn. So please join us in that edition as well. So that's all on my part. I don't know if Jason want to add something.
Yeah, I want to thank everyone again for joining us today, and thank you to our moderator, host, and especially to all our presenters who presented their materials today. As Pablo said, there's a part 2 in the next few weeks, but know that this webinar is recorded, and once all of the recording are ready, we'll post it and have it accessible online. So keep an eye out for the recording, but also hope we'll see you in a few weeks for the next part. Thank you, everyone, and I hope you all have a great rest of your week. Bye, everyone.
Bye, everyone.